Today, we're diving straight into the incredible engineering of the cardiovascular system, anchored entirely by a life-or-death clinical case. We're going to explore how the human heart and blood vessels function not just as some abstract concept, but as a dynamic, living machine that can, and sometimes does, falter under pressure.
Here's our roadmap for today. We'll start with a clinical case on dyspnea, then look at the origins of the heart, the biology of blood vessels, the anatomy of a heartbeat, dive into preload, afterload, and contractility, and finally, we'll solve that clinical case.
Section 1. A clinical case. Dyspnea.
So picture this. A patient is just trying to take a normal, everyday walk. But instead of their heart easily matching their body's increased demand for oxygen, their lungs begin to fill with fluid. It's a terrifying condition known as pulmonary edema, leading to severe dyspnea or shortness of breath. We need to figure out why their biological system is failing them at such a critical moment. Normally, when we exercise, our heart responds beautifully. As the heart muscle stretches, it pumps harder to meet the demand. But for our patient, it's a completely different story. Their failing heart muscle, the myocardium, just can't increase its contractile squeeze. So instead of safely handling the activity, the heart just stretches more and more, pushing them straight into the danger zone of pulmonary edema. The pump is literally overwhelmed.
Section 2. Origins of the heart.
To figure out why the pump fails, we actually need to rewind all the way back to embryogenesis. It's wild to think about, but the heart starts as this simple, single-layered tube. Then, it undergoes this complex, asymmetric looping, basically twisting and folding in on itself, to create the distinct atria and ventricles we rely on. Different parts of the mature heart actually develop from totally different populations of embryonic cells. And that totally explains why certain congenital diseases only target specific areas, like the right ventricle or the outflow tract.
Section 3. Biology of blood vessels.
If the heart is the pump, then your blood vessels are the pipes, and their structures are drastically different depending on what they need to do. Down at the micro level, you've got these super thin, single-layer capillaries, built for fast exchange of oxygen and nutrients. But zoom out to the macro level, and you find thick, muscular, and elastic arteries. These larger vessels have a structured middle layer called the tunica media, packed with smooth muscle cells and elastin. This lets them act almost like rubber bands, storing the kinetic energy of a heartbeat and tightly regulating the exact blood pressure our struggling patient is failing to manage.
Now here is the really crucial part. When the endothelium, that's the lining of these pipes, becomes dysfunctional, it completely flips its behavior. It goes from being a protective, anti-inflammatory barrier to a leaky prothrombotic one, meaning it suddenly becomes very prone to dangerous blood clots. Normally, these cells release nitric oxide to keep blood flowing smoothly, but under severe stress or disease, the barrier just fails. That impaired permeability leads straight to conditions like capillary leak, which is exactly why our patient's lungs are filling with fluid. The pipes are literally weeping fluid into the surrounding tissues.
Section 4. Anatomy of a heartbeat.
Let's zoom all the way into the microscopic engine itself. A cardiac muscle contraction is this incredibly precise four-step mechanical rowing motion, all powered by cellular energy. It's a continuous, relentless cycle of tension and release, happening billions of times over your life.
So, how does it work? Thick myosin filament heads basically grab onto thin actin filaments and pull. First, ATP is split to give the myosin head its energy. Then calcium sweeps in to expose the binding sites. After that, the products of that split ATP are released, and boom, you get the mechanical power stroke, the actual contraction. Finally, and this is super important, without a brand new molecule of ATP binding to that complex, the muscle literally cannot relax for the next beat. Actually, a little morbid fun fact for you, this exact lack of ATP is why a body gets stiff after death, a state literally known as rigor mortis.
But what's actually conducting this whole molecular orchestra? It's calcium. When the heart gets an electrical signal, a tiny bit of trigger calcium enters the cell from the outside. That little trigger tells the cell's internal storage unit, the sarcoplasmic reticulum, to dump a massive internal store of calcium. We call this a calcium spark. It floods the system, binds to troponin C, and forces the muscle to contract. Then, to relax, molecular pumps have to aggressively sweep all that calcium right back out of the way.
Section 5. Preload, afterload, and contractility.
All right, let's zoom back out to the entire organ. There are three master variables that completely determine how much blood your heart can pump with every single stroke. First is preload. Think of preload as how much blood fills and stretches the heart right before it beats. Second is afterload. That's the resistance the heart has to push against, like the blood pressure in your aorta. And third is contractility, which is just the raw, inherent squeezing power of the muscle fibers themselves.
When you put it all together, stroke volume and heart rate dictate your total cardiac output. And that output, combined with vascular resistance, tightly regulates your arterial blood pressure. Your body is constantly monitoring this pressure using baroreceptors in your neck. They send rapid-fire feedback loops to your brain to instantly tweak your heart rate or constrict your blood vessels to keep everything perfectly balanced. It is honestly a masterpiece of physiological systems engineering.
Now, there's a simple rule of physics at play here. In a pressure-volume loop, the width of the loop represents the stroke volume, the actual amount of blood ejected. If you raise the afterload, meaning the heart has to pump against much higher pressure, the peak pressure shifts up and the width of the loop shrinks. The stroke volume drops and more blood gets trapped inside the ventricle. And if contractility drops, you get the exact same bad result, a weaker squeeze, less blood pumped out, and a dangerously backed up system.
Section 6. Solving the clinical case.
Armed with all this biology, let's go back to our patient, who is gasping for breath on their walk. A healthy heart would just use adrenaline to instantly ramp up contractility and handle the extra blood returning from exercising muscles. But our patient's diseased heart? It just can't. Its inherent contractility is broken. So the heart gets completely overwhelmed by the preload, the sheer volume of returning blood. Because it can't push that blood forward against the afterload, the pressure inside the heart spikes. That pressure backs up directly into the pulmonary capillaries, destroys their selective permeability, and violently leaks fluid right into the lungs.
And that brings us to a fascinating final puzzle. If you were the doctor standing right at their bedside, which part of this complex cardiovascular machine would you target first to pull them back from the brink? Would you give a diuretic to drain the fluid and lower the preload, maybe a vasodilator to drop the resistance and ease the afterload, or a medication to directly supercharge those calcium sparks and boost contractility? The beauty of understanding this biology is that it gives us the exact tools to answer that very question.